and welcome to Spotlight on Antibodies, a podcast from BioIntron where we bring you the latest antibody industry trends. This month in May, we'll highlight the future of VHH technology, chemical conjugation in antibody therapeutics, and emerging trends in antibody characterization. Now let's begin with VHH antibodies, also known as nanobodies. Most people are familiar with conventional monoclonal antibodies, or IgGs, which have become an important class of medicines across oncology, autoimmune disease, and other therapeutic areas. VHHs are different. They are single-domain antibody fragments derived from camelid antibodies, the type naturally found in animals such as llamas and alpacas. Because VHHs contain only one antigen binding domain, they are much smaller than a traditional IgG antibody. That smaller size can give them several useful properties, including good tissue penetration, access to recessed or difficult-to-reach epitopes, and stability under conditions that may be challenging for larger antibody formats. In simple terms, VHHs are small antibody binders that can sometimes reach spaces where conventional antibodies may have limitations. One area drawing attention is the shift from traditional discovery towards more intentional design. Historically, antibody discovery has relied on screening large libraries of candidates, essentially searching through many possibilities to find the few molecules that bind well. For VHHs, this has included approaches such as animal immunization, synthetic libraries, and repeated rounds of selection. Increasingly, researchers are adding computational design and artificial intelligence into that process. Instead of replacing, these tools may predict how a VHH binds to its target, identify mutations that improve stability, reduce aggregation, or support human compatibility, and flag potential developability issues earlier. For example, VHH-specific tools, such as NanoBERT and the therapeutic nanobody profiler, have been developed to better account for the structural and sequence features that distinguish nanobodies from conventional antibodies. Another reason VHHs are interesting is that they can be linked together into larger engineered formats. This makes them useful as building blocks for multi-specific therapies, which are molecules designed to bind to more than one target, or more than one site on the same target. In oncology, VHHs can act as therapeutic binders on their own, but they can also serve as targeting modules for drug delivery, radionuclide delivery, imaging agents, or immune cell platforms such as CAR-T, CAR-NK, and CAR macrophage systems. Researchers are also using strategies such as peptide linkers, hinge domains, self-assembling systems, and chemical conjugation to build multivalent or multispecific VHH constructs. These designs may improve apparent binding strength through avidity effects, increase specificity, or enhance functional stability. Of course, for any therapeutic platform, the scientific promise has to be matched by manufacturability. VHHs can be produced in several expression systems, including mammalian cells, bacterial, or yeast, and even plant-based systems. This flexibility supports scalable production, though each product still needs its own development and manufacturing strategy. At the same time, VHHs have known limitations as their small size, while useful for tissue penetration, can also lead to rapid renal clearance, which means they may leave the body quickly. Researchers are addressing this through approaches such as fusion to larger proteins, or chemical modifications like pegylation to extend circulation time. Thus, the future of VHH technology will be about using their structure in a more programmable way, designing them with computational tools, assembling them into multi-specific formats, and adapting them for therapeutic, diagnostic, and delivery applications. The idea of engineering antibodies into more functional formats brings us to our second topic, chemical conjugation in antibody therapeutics. Chemical conjugation refers to the use of defined chemical reactions to attach another molecule or payload, to an antibody. That payload might be a cytotoxic drug, an imaging agent, a radionuclide chelator, an oligonucleotide, a peptide, protein, or even another antibody fragment. The antibody provides target recognition, while the payload adds a therapeutic or diagnostic function. One of the best-known examples is the antibody-drug conjugate, or ADC. In an ADC, an antibody is chemically linked to a potent drug. The antibody helps guide the drug towards cells that express a specific target antigen, while the attached drug provides the cell-killing activity. But one of the long-standing challenges in ADC development is their control. Early conjugation methods used naturally occurring lysines or cysteines on the antibody surface. These approaches can generate heterogeneous mixtures, where payloads attach at different sites and in different numbers. That matters because the drug-to-antibody ratio, or DAR, can influence potency, stability, pharmacokinetics, and tolerability. This is where site-specific conjugation becomes important. Site-specific conjugation means attaching the payload at a defined location on the antibody. In principle, this can improve product uniformity, preserve antigen binding, reduce aggregation, and make the final molecule easier to characterize and optimize. One example is the AgiCap platform reported by Fuji and colleagues. This approach uses Fc affinity guided chemistry to modify native antibodies at defined lysine residues in the Fc region without requiring antibody engineering. In their second-generation version, the process was simplified by removing redox treatment and enabling a more streamlined one-pot modification workflow. The researchers generated more than 20 ADCs using different antibody and linker payload combinations with low aggregation. This work also highlights a point that is especially relevant for ADC design. More payload is not always better. A higher DAR may increase drug loading, but it can also increase hydrophobicity, reduce solubility, promote aggregation, and affect pharmacokinetics. Homogeneous DAR2 ADCs, where 2 drug molecules are attached per antibody at defined Fc sites, may offer a balance between potency, tolerability, and stability. In the AgiCap study, site-specific trastuzumab MMAE ADCs showed anti-tumor activity in a HER2-positive xenograft model, while pharmacokinetic and tolerability data supported the potential for an improved therapeutic index compared with more traditional stochastic ADCs. Chemical conjugation is also being explored as a way to modify antibody biology. In another study, researchers attached an engineered peptide to the Fc region of trastuzumab. The peptide carried an azide group, which could then be connected to DOTA, a chelator used to bind diagnostic or therapeutic radionuclides. In other words, the antibody first received a peptide adapter, and then a payload could be added through click chemistry. Click chemistry are fast, selective reactions that can connect molecular components under mild conditions. For antibody therapeutics, that is useful because the reaction needs to be efficient without damaging the antibody structure or binding function. Interestingly, the peptide and peptide DOTA conjugated trastuzumab showed enhanced antibody-dependent cellular cytotoxicity, or ADCC. ADCC is an immune mechanism in which immune cells, especially natural killer cells, recognize the Fc region of an antibody bound to a target cell and help destroy that cell. The study found stronger Fc gamma receptor 3A binding compared with unmodified trastuzumab, which helps explain the improved ADCC. Structural and thermodynamic analyses suggested that the peptide may act like a molecular wedge between Fc domains, reducing Fc flexibility. That reduced Fc dynamics was associated with both enhanced effector function and improved thermal stability. A third direction is the use of bioorthogonal chemistry to assemble bispecific and multispecific antibody formats. Bioorthogonal means the reaction is selective and compatible with biological molecules, so it can occur without interfering with native protein function. Recent research demonstrated fluorescent bispecific Fab conjugates, as well as bispecific ADC-like constructs that combined antibody fragments with the payload MMAE. This type of modular chemical assembly could be useful for early-stage screening, especially in bispecific ADC development. For these molecules, researchers may need to test many variables at once, and a modular conjugation strategy could make it easier to swap components and compare designs without rebuilding the entire antibody each time. Taken together, these studies show how chemical conjugation is moving beyond just a payload attachment as a site-specific ADC production, Fc-directed peptide conjugation, and bioorthogonal bispecific assembly point towards a more controlled and adaptable antibody format. Let's talk about characterization. Binding specificity is one of the most important characteristics of an antibody drug candidate. However, antibody characterization is multidimensional, combining structural analysis, biological activity, immunogenicity assessment, and functional outcomes. To understand why this matters, it helps to briefly revisit antibody structure. A conventional antibody is a Y-shaped glycoprotein made of 2 heavy chains and 2 light chains. The Fab region, or fragment antigen binding region, is responsible for recognizing the target antigen. The Fc region, or fragment crystallizable region, interacts with immune components and can influence mechanisms such as immune cell engagement, complement activation, and antibody half-life. But beyond specificity and affinity, antibodies carry molecular features that can influence how they behave in the body. One example is post-translational modification, or PTM. These are chemical or structural modifications that occur after the antibody protein is made. Glycosylation and oxidation are 2 common examples. These modifications can create molecular heterogeneity, meaning that even one antibody product may contain several related molecular variants. That heterogeneity can affect stability, pharmacokinetics, immune cell engagement, and potential immunogenicity. Antibodies can work through several mechanisms. Some directly neutralize the target. Others activate immune-mediated cytotoxicity. Some modulate receptor signaling pathways. To evaluate these effects, researchers use cell-based assays and functional bioassays, which measure outcomes such as cytotoxicity, receptor activation, signaling inhibition, or complement activity. These assays are important because binding measurements alone do not always predict biological activity. 2 antibodies may bind the same antigen with similar affinity, but still differ in Fc function, signaling effects, internalization behavior, or immune engagement. Advanced analytical technologies are helping researchers connect these layers of information. Mass spectrometry can support sequence confirmation and PTM analysis. Chromatography methods such as HPLC, size exclusion chromatography, and ion exchange chromatography help separate and quantify variants. Spectroscopy methods, including NMR and FTIR, can provide information about higher-order structure, the 3-dimensional conformation of the antibody. The larger trend is integration. Researchers are increasingly trying to connect molecular structure with biological function. So antibody characterization is like a connected framework where structural analyses tell us what the molecule is, binding studies tell us how it interacts with the target, functional assays tell us what it does in a biological system, and immunogenicity assessment helps evaluate how the body may respond to it. The more complex the molecule, the more important it becomes to understand not only its structure, but also its behavior across biological and manufacturing contexts. And that's all for this month's Antibody Industry Trends. To learn more insights into antibody drug news, make sure to follow BioIntron on LinkedIn or our website at www.biointron.com. Keep up to date with further episodes from Spotlight on Antibodies by following this podcast.